Hello everyone, my name is Mario. I'm a PhD student in the Department of Mechanical Engineering here at UNLV. Today I would like to talk about a research work which is currently under preparation. In this work, my team and I have been adding surfactants in boiling water. So let's get started. In my research, I deal with large amounts of vapor bubbles. These bubbles are generated on a heating surface that is removing heat. This heat transfer process is known as boiling. Boiling is ubiquitous, meaning it can be found in electric kettles, in power plants, or even in avionic components. Boiling is important because it's a very effective mechanism to remove these vast amounts of heat. However, to date, improvements to boiling performance have been very challenging due to the unpredictable and complex nature of bubble behavior. In the boiling community, we usually want to quantify the enhancement. One way to quantify enhancement is by looking at the heat transfer coefficient, which is the HTC, the ratio of the heat flux over the wall superheat. Looking to a plot having the heat flux as a function of the wall superheat and having a reference boiling curve, typically the boiling curve of water, we know we have enhancement or we have improved the boiling curve when this is shifted to the left or upwards, meaning that at the same wall superheat we have a higher heat flux. So there are many ways to improve boiling. One way to improve boiling is by adding surfactants in boiling water. Surfactants are tiny molecules that they dissolve in water, they absorb to the interfaces, they alter the interfacial properties of the liquid. A surfactant has two components, a hydrophilic head and a hydrophobic tail. It has been known that increasing the surfactant concentration that improves the heat transfer coefficient or lowering the wall superheat. However, we don't know exactly what amount of surfactant we need to add in the solution. So to better understand surfactants, we need to also look to another property of surfactants, which is the CMC. The CMC is the critical micelle concentration. At concentration lower to the CMC, the absorption at the interface or interfaces begin. At or near the CMC, the absorption stops and the surfactant monomers are just swimming around the solution or the boiling water. At concentrations above the CMC, also the absorption was stopped, but the surfactant monomers form micelles, and this process is known as micellization. So we have been trying to understand if there's a concentration linked that we have to look into the CMC of every surfactant. So therefore, this arrives to the question that what concentration surfactant will provide an optimal enhancement, right? So previous studies have suggested that the optimal concentration, that the optimal enhancement should occur near or at the CMC. The hypothesis here is that there should occur separate optimal peaks for a wide range of surfactants. Surfactants with low CMC, surfactants with a higher CMC, and this is for the case where the absorption and micellization equilibrium case occurs. Okay, so how do we test this hypothesis? All right, so in our lab, we built a custom boiling device that had a heating surface oriented in the vertical position, and we added surfactants. For each surfactant, we conducted 10 to 15 concentrations, and each concentration was carried out 3 times or more. And these amounted over 200 boiling experiments. Have a look at this video. It's really, really cool. This is in a heat flux around like 20 watts over centimeter square, which is a lot of heat being dissipated. This is in boiling water. Okay, so what our experiments discover? Let's have a look. Surprisingly, if you remember, the hypothesis that we should see the optimal HTC around the CMC of the surfactants for every surfactant. For the surfactants with this low CMC, the optimal HTC occur in a range from 1 to 10 millimolar. Alright. For this other surfactant, that optimal HTC occurred near to its CMC value. That was a coincidence. Also in the range from 1 to 10 millimolar. For these other two surfactants that have a higher CMC value, the optimal HTC also occur in the range from 1 to 10 millimolar. So here we discover there is an optimal concentration range, and this led us to the statement to know that we were wrong. Yes, the hypothesis that I just mentioned in the previous slides, it's wrong. The separate optimal peaks hypothesis is not valid. Therefore, there is a universal concentration range of enhancement which is independent of the CMC. And yes, and the equilibrium absorption micellization case does not look appropriate in this case. Alright, so this all sounds really cool, but how can we explain this? One way to explain this is by conducting a surfactant mass transfer analysis in a control volume region, by equating the incoming advected surfactants with the departing absorbed surfactants. This value of gamma, which is the surface concentration of the absorbed surfactants, does not change in order of magnitude for all these wide range of surfactants. Also, the bubble departure radius also doesn't changes in order of magnitude. By solving for a balanced concentration, plugging the values of gamma and the bubble departure radius, and keeping constant the values of the liquid and vapor densities, we found there is a balanced concentration, which is in the order from 1 to 10 millimolars, which is in great agreement with our experimental data. So here this suggests that this is not an equilibrium problem, as how we thought at the beginning, and this might be a mass transport limited behavior. So when we were conducting these experiments, we saw the HTC reach a peak, right? So if you remember here are all the peaks in the optimal concentration range. So we were wondering why the HTC degrades. We conducted some viscosity tests, and we're thinking with what I just mentioned, that right after reaching this optimal, why the HTC does not stabilize, but we saw in our case the HTC just degrades. We saw in literature there is a relation of the HTC which is proportional with the viscosity, and therefore we conducted an experiment of, with another custom device that refabricated in our lab, and we tested the viscosity for this specific surfactant, S12S, in a room temperature condition. We saw that at concentrations higher to the around 10 millimolar, the HTC increases, suggesting that the idea, like if you remember, the optimal concentration range is from 1 to 10 millimolar, and after that the HTC degrades. So this is in a great agreement, suggesting that the HTC is a viscosity-induced degradation. Alright, so to sum up, we have three key points here. There is an optimal concentration range which is regardless of the CMC value. That optimal concentration is linked to a mass transfer balance with the incoming advected surfactants and the departing absorbed surfactants. And remember, very high concentrations decrease the HTC, and that's due to the increase in the viscosity. Our future work will be focused on doing detailed visualization that will help us to conclusively determine how these surfactants enhance boiling heat transfer. This work wouldn't have been possible without the help of my beautiful boiling team and the collaborators from MIT. I would like to thank to my academic advisor, Professor Jeremy Cho, who supported and financed all this work. If you have any questions or suggestions, please contact us at the Kind Lab, or feel free to email us to our corresponding emails. Thank you for your attention.